Okay, our next speaker is Frank Criscione.
Alrighty. So good afternoon, everyone. My name is Frank Criscione. I'm a postdoc at the University of Maryland Institute for Bioscience and Biotechnology Research, and I currently work in the lab of Dr. David Brogda. Mainly, we work on mosquito transgenics, but my project focuses mainly on mosquito hemocytes and the role that they play in the insect immune system. So, most of you know, and as Alvaro made a much prettier picture than I did, about how mosquitoes initially get infected with Plasmodium. First, they have to uptake an infected blood meal. When they do that, the gametocytes actually go into the midgut and then mature into oocysts. Once they mature into oocysts, they egress the midgut and actually form sporozoites. So what you see here is, there we go. So these are the sporozoites. The sporozoites then go and infect the salivary glands, and then that's how a mosquito can go and transmit malaria and all these other diseases to humans. But the mosquito doesn't just sit there and do nothing about sporozoites. It actually mounts a defense. And that defense is done by the hemocytes and other factors, but these are the cellular components to that defense. And hemocytes are broken up into three classes. There are nosotoids, granulocytes, and prohemocytes. And these are like I said, the cellular defense. So I really wanted to know what the main job of these hemocytes is. And actually, a lot of people in this room have studied hemocytes and their role in insect immunity, and I thank you all for doing that. But what a lot of people have done is they've used injection of double-stranded RNA to get at the molecular questions involved in hemocytes. And when injecting double-stranded RNA into the mosquito, what you actually wind up seeing is that you're going to have effects in multiple tissue types. You're going to affect the midgut, you're going to affect the fat body, you're going to affect everything, including the hemocytes. But if we really want to get at what's actually happening in the hemocytes molecularly, we have to affect only the hemocytes. So that means injecting double-stranded RNA is not going to be a solution for us. So, and why are we even worried about that? Well, hemocytes are actually really important. They actually deal with about up to 80% of the parasitic infection. And they do this by melanizing parasites, performing lysis, and phagocytosis. So understanding the molecular mechanism is, like I said, an extremely important thing. But we had to create a tool to do this. And the tool that we decided to create was an enhancer-based Gal4 UAS system. So what we did here was we took a Gal4 cassette. We have multiple lines in our laboratory, and we have a Gal4 expressing line, of which we then remobilized throughout the entire genome of the mosquito. And through this screen, I screened about 60,000 individual larvae to detect enhancers. So what happens is during this remobilization event, this Gal4 cassette is going to land in a new spot on the genome, and if it lands near an enhancer, and then we cross that line with a UAS TD tomato, that Gal4 will be expressed in the pattern of that enhancer, and then TD tomato, which is a red fluorescent protein, will be expressed in the pattern of that enhancer. So, like I said, after screening 60,000 larvae, we found 3 lines that just express the TD tomato in hemocytes and no other tissues. Now, mind you, I did find about 15 other lines that express hemocytes, expressing hemocytes and in also other tissues, but those are pretty much useless to us because we said, as I said, it would be pretty much just like injecting double-stranded RNA into the entire mosquito. And as you can see here, I guess the lights are on, so it's a little bit hard to see. But this is one of the larvae, and you see these little red dots. Those are all hemocytes. And then this is an adult female mosquito, and through the cuticle, or where the membrane is thinnest, you can actually see some of the little red dots also over there. So, great. We can turn hemocytes red. Kind of useless if you're studying immunology. We really want to get at what is happening to these hemocytes. So again, just to confirm that I wasn't going crazy and we just didn't turn random dots red in the larva, we did immunofluorescence or indirect immunofluorescence to see that yes, we are preferentially staining hemocytes because here's wheat-germ agglutinin that stained the hemocytes, and then we also have an antibody to TD tomato, and that all co-localized in this granulocyte. So great. Now we have the tool. to express something specifically in a hemocyte. What do we want to look at? And we decided to target the IMD pathway. Now we know that the IMD pathway is involved in defense against Plasmodium. So what happens is that a parasite will send a signal once it basically an extracellular signal, which starts the IMD pathway cascade cytosolically, and that causes cleavage of REL2. Cleavage of REL2 then goes into the nucleus, and then that starts the transcription of effector molecules, which then are responsible for going out and dealing with this parasitic infection. So Caspar is also listed here because Caspar is a negative regulator of the IMD pathway. That means when there is no infection present, Caspar is expressed and it blocks the IMD pathway. When an infection is present, Caspar is then blocked, which then allows the IMD pathway to proceed in its expression and then creating effector molecules which go out and fight the pathogen. So what we created were two constructs that were going to be mixed with our our hemocyte enhancer line expressing GAL4. So what we did is we crossed our hemocyte line with a UAS Caspar overexpressor. And what you see here in wild type, this is actually RNA that's isolated from hemocytes 7 days post a Plasmodium burgii infected blood meal. And what you see here is that we do actually see an increase in Caspar, meaning that that despite the infection, we are keeping Caspar active. So normally that's not the case. If there is an infection present, Caspar would then be shut off. And then here what you see is that we are having basically the opposite effect, a knockdown. So in the event that despite an infected blood meal, we are actually causing a knockdown that's greater than what is normally expected in these animals. So we're basically looking at the IMD pathway in two forms, on or off, despite the presence of parasites or not. So when we actually perform a parasitic blood meal, and let me just talk about the cross a little bit more. Here what we actually are doing is we generate heterozygous crosses. So our GAL4, which is the hemocyte-specific GAL4, is a heterozygote, and here our UAS is a heterozygote. Why do we do that? We do that so that when we actually perform the cross, we then generate all four genotypes, meaning that we get a wild-type genotype, we get a GAL4-only genotype, we get a UAS-only genotype, and then we get the experimental genotype, which is the GAL4 and the UAS. We can separate these all by looking specifically at the colors that they produce. So our GAL4s have 3xp3 blue, meaning that their eyes are blue, and our UAS is green. So it's 3xp3 green, their eyes are green. So anyway, going on to that. So when we looked at the knockdown, what we actually saw was that there was a slight decrease in the number of oocysts present. So this, in a way, makes sense. If you knock down Caspar, you're ramping up the mosquito's immune system before it even sees a parasite. So if a parasite is then, seen in the mosquito, then the immune system would have an advantage in responding against it, and therefore we see a total number of oocysts that are less. This is significant against the wild type, and then when you actually look at sporozoites, and I will say that the Plasmodium burgii that we are using is a GFP luciferase expressing form of the parasite, and I like luciferase assays, which is why I picked that strain. So when we were looking at the sporozoite invasion of the salivary gland, that was our kicker in saying, because originally we thought, all right, the hemocytes have to stop the sporozoites from getting to the salivary gland somehow. So we were going to look at the sporozoites in the actual salivary gland, and what we see is a pretty dramatic reduction of the total sporozoites in the salivary gland in comparison of our cross and the wild type. Now, I'm not blind. There is a reduction in the other ones as well, but that could be due to position effects in our genetic schemes that we were using. So that's why we actually generated all of the four genotypes to actually see if there were position effects that could lead to having a more resistant strain of the mosquito. Now, we then decided to do the opposite, and this is the more striking example, because what we saw was that when we leave Caspar on, meaning that Caspar is on and the IMD pathway is permanently shut off in hemocytes only, what we saw was that you did have infection in all of the other 3 genotypes, but then in the cross, you have a much higher infection, and that is recapitulated in the sporozoite counts. So when we're looking at this, this is in some ways really saying that yes, the IMD pathway is very important when it comes to hemocytes, and specifically in the sense of both oocysts and sporozoites. And when originally our hypothesis was that it was really only going to affect the sporozoites. So we wanted to figure out molecularly why is this happening. So we decided to look at those downstream effector molecules, APL1 and TEP1. And in our overexpressor, we actually saw that there's no difference between wild type and our cross. There's no difference in the expression between an APL1 or TEP1 in the overexpressor, meaning that just because the IMD pathway is shut off doesn't actually mean, because, for instance here, the IMD pathway is not actually shut off in the wild type. It's only shut off in the cross. So why is it that APL1 and TEP1 are just not affected? But then when we look at the Caspar knockdown, again, in the knockdown, we see that APL1 is not really that, it is upregulated, but TEP1 is greatly upregulated. So that means that when we knock down Caspar, leaving the IMD pathway on the entire time, that TEP1 is capable of building up in the mosquitoes, in the hemocytes, and potentially causing that small decrease in the number of oocysts and sporozoites that we see. So let's put that all together. And so now we have a little bit of a new hypothesis based on the data that we acquired. So with Caspar being knocked down, we see fewer oocysts. We see fewer sporozoites. And we see an increased amount of TEP1 being expressed. And we see a slight increase in APL1. When we overexpress Caspar, what we then notice is that there is a great increase in the number of sporozoites. There's an even higher increase in the number of oocysts, or there is a higher number of oocysts and a higher number of sporozoites. And we see that there is minimal change in TEP1 and APL1. So now this lends us to believe that hemocytes may contribute to the defense against infection at the oocyst level in a fashion that is dependent on the IMD pathway being activated, and that the ectopic activation of the IMD pathway in hemocytes may contribute to the reduction of infection through increased expression of TEP1. So one of the main reasons why I think that this is really fascinating to look at is because this is one of the first times where we're capable of targeting just the hemocytes. So that means that previously all the data that we have could have been combined effects of fat body, midgut, and everything else influencing the responses that we see in hemocyte molecular biology. So this is, again, one of the first times that we're capable of seeing just what the hemocytes are contributing to the defense of the Plasmodium infection. So with that, I'll just like to thank my fellow lab members, my PI, and also Andresa Chagas. She helps a lot with my dissections and preventing me from freaking out when I don't understand a bit of data. And also funding and all of you guys for paying attention to me for the last 13 minutes.
We have time for questions.
Very nice talk. So I'm a little bit confused because there are several studies now from several groups using RNAi and transgenics overexpression of REL2 in the fat borne in the midgut that have shown that the IMD pathway is not very active against Plasmodium berghei.
Right.
So I'm wondering whether your results really show a very specialized role of the hemocytes in the defense against this particular parasite species. You should really do experiments with falciparum, I think.
It's funny that you mentioned that because we are working with you to do the experiments with falciparum. But yes, this is one of the things that we were interested in because, like I said, when I first did this experiment, it was quite shocking. Berghei doesn't really respond. The IMD pathway is not really responsible in mounting the defense against Berghei. And so when we saw that there was a response, we said, okay, well, how does this then compare to falciparum? But we are currently working on those experiments. We're just having a few technical issues, but we're working on it.
Thank you. Great talk. Very cool project. Quick question. So when you did your crosses, get your 4 genotypes out, you infect them, and then you check for sporozoites in salivary glands. Obviously, your combination line has the lowest sporozoites, and then the next lowest one is basically with your double-stranded RNA on its own, correct? It's the UAS Caspar knockdown on its own.
Right,
yeah.
right.
And that's probably position effect, a little bit of leakiness, right?
Yeah, yes.
So
yeah.
my actual question is, how long have you had those lines? Because there's been kind of rumors that if you have transgenes expressing double-stranded RNA, that the mosquito does kind of epigenetically silence it over time, and you kind of lose the effect. So have you seen that? Can you comment on that?
Right. So that's one of the actual benefits of using the Gal4 UAS system, because we keep these lines separate. The overexpression doesn't happen until a cross occurs.
But yours is suggesting you've probably got leakiness, which is where the concern arises,
okay,
right?
So
yeah.
Good point. That's all I'll say.
Oh, okay. Okay. Well, I didn't know how long you've had the lines.
So the UAS lines we've had for about 2 years now, and the enhancer lines we've had for about a year. But that is a very valid point. That yes, that